I've known him for 20 years, time flies, when we're having fun. And he's an expert in hyperpolarized, various hyperpolarization technique, including dynamic nuclear polarization, as well as the hydrogen-based hyperpolarization technique. So Aaron is the director of preclinical MRI core at the Beth Israel Deaconess Medical Center. Aaron received his PhD from University of Chicago doing high energy particle physics. And after postdoc studies at UCLA and Harvard, he transitioned to MRI research. And since then, he's done amazing work on various cancer models using hyperpolarized imaging technology. And more recently, he developed a new hyperpolarized contrast agent to look at tissue perfusion as well as in animal as well. And the image is stunning. It's amazing. So that's the topic he's going to talk about today. Without further ado, let's welcome Dr. Grant.
So thank you, Dr. Yen, Dr. Yu, for the kind introductions, and thank you for the invitation. So I'm going to be talking today about some quantitative imaging work that I've been doing, mainly in the context of perfusion. Many of you may have seen some of this last July when Yifen organized the workshop here on X-nuclear imaging. Today it's going to be an expanded version of that, and hope to say a little bit more about how this fits into the big picture of hyperpolarized carbon-13. So the slides will advance. There we go. All right. So very, very briefly, I'll say a few words about hyperpolarization technology. I'm sure many of you know this. Talk about some common techniques that people are currently using for metabolic quantitation with pyruvate. Talk about how complex these signals are in principle. I'm sure many of you have thought about this and realized that there's really a lot going on there. And then we're going to turn our attention to a much simpler case where you don't have to think about metabolism and transport and uptake and all that. We're just going to think about perfusion imaging with a very simple tracer that we developed for this purpose. Talk about mapping correction for T1 and T2 decay, modeling of the tracer kinetics, and determination of absolute blood flow in tissue. I'll say a little bit about CSF, and I should say that I hope that if you have any thoughts about what's going on with the CSF, that you will educate me. I'm mainly a C13 guy who's sort of dabbling in perfusion rather than the other way around, and so I'm sure there's opportunities for this audience to teach me, and I hope that you will. And then I'll say a few slides at the end about how we hope we might be able to extend some of these methods, both the high resolution imaging as well as some of the quantitation methods to pyruvate. So just a few preliminaries about the technology. Of course, as I'm sure most of you know, carbon-13 is a wonderful molecule, a wonderful nucleus for probing all kinds of metabolic pathways. It occurs in all kinds of metabolically relevant compounds. But sensitivity has always been the major limitation, and imaging with this stuff is just impossible. And so hyperpolarization, of course, is a technology that takes the basically randomized spins and seeks to align them as much as possible. And you can get a boost in signal from a substrate like this by a factor of 10,000, maybe 100,000, depending on your field strength. And now you can do sort of real-time spectroscopic imaging and metabolism. And there are two techniques for doing this, and I'll just briefly mention those: dynamic nuclear polarization and parahydrogen-based methods. So dynamic nuclear polarization is the most broadly used technology right now. The idea is you take your substrate and basically by brute force, you freeze it about 1 degree above absolute zero, put it in a strong magnetic field. You freeze it in the presence of a radical, which has an unpaired electron. And that's important because even at these low temperatures and field strengths, you still don't get very much carbon-13 magnetization. So you need that electron and its huge gyromagnetic ratio to get a species of spins in the sample that's essentially 100% polarized. The electrons are 100% polarized. And then you apply microwaves, and through sort of an analog of the nuclear Overhauser effect, you push that polarization onto the carbon-13. And when you're ready, you basically inject. You've got your sample here frozen under liquid helium. You're putting in your microwaves, and you've got a line where you inject some hot saline and dissolve your sample. You get it out, quickly transfer it, inject it, and image it. So that's DNP. It's relatively slow and expensive, quite expensive, but it also offers you a flexible choice of substrate. All you really need to do is be able to mix it up with a radical and get it to freeze in a glassy state. So parahydrogen is a really beautiful technology that Matt Rosen and others over here are interested in. Parahydrogen gas is basically the nuclear spin isomer of hydrogen that you get when you just cool the hydrogen down to about 10 or 20 Kelvin and then warm it back up. In parahydrogen, you're in the nuclear spin singlet state, meaning that the two nuclei in the hydrogen molecule are oppositely aligned. And what that means is that there's a very high degree of nuclear spin order here, although there is no polarization. This gas is wonderful because you can take it back up to room temperature. You can store it for months. And in fact, I used to dabble in parahydrogen, and this sort of $50 of stuff from Sigma and Home Depot, you can basically do this, in the lab. You don't get huge polarization, but you can do it. So it's a very easy, very fast technology. The way that you get this nuclear spin order into magnetization is by doing either hydrogenation. You add the parahydrogen across a double or triple bond in a carbon-13 labeled molecule, and that can push the polarization onto carbon-13. There's also strategies here that Matt Rose and another are interested in, where you don't actually do a chemical addition, but you just associate with an iridium center, and in that way you can transfer the polarization. But it's a very beautiful technology, and I should say that Yifan Yen and Leo Tsai have been working with Envision to get, this is a preclinical polarizer, but there's a clinical parahydrogen polarizer in the works that's potentially a very exciting technology, because this is a fast, cheap way of doing hyperpolarization relative to DNP. You can sort of do one injection after another. You don't have to wait for hours and hours. So pyruvate metabolism, of course, is the big one for most of hyperpolarized C13. So glucose metabolically gets broken down into two pyruvates, and then you have various disease states that modulate the lactate signal relative to pyruvate. I'm not going to belabor the point, but basically when you inject this pyruvate, you're looking at these downstream metabolites and looking for how that might be a diagnostic biomarker. And so you inject the stuff, and here are some a series of spectra acquired with this tracer. Initially, you see just the pyruvate. Sometime later, you pick up all the downstream metabolites. This is about 20-25 seconds later, and you see the lactate, pyruvate, hydrate, alanine, bicarbonate, and so forth. So you can pick all of these up and monitor them more or less in real time. Pretty easy to do. Now, what about quantitation? And at this stage, so this is a paper that Leo Tsai was last author on. I worked with him, supplied the C13 data for this one, where we were looking at hepatocellular carcinoma and sort of stratifying different types of HCCs based on their underlying genetics in terms of what happens with hyperpolarized carbon-13. And what they found is that sometimes, but not always, you will see a high lactate signal in these tumors, but that isn't true across every single strain of HCC. And you see these kind of time courses where you have the pyruvate and lactate signal, and now you want to be able to put a number on it. And people use various things. You can use the area under the curve, so the area under the lactate curve relative to pyruvate, or you can do some kinetic modeling like this, where you've got basically a rate constant for pyruvate being turned into lactate, simple first order differential equation. But you can imagine that there's an awful lot being rolled into that equation. So we've said there's a rate constant here, but of course there's T1, T2, wash in, wash out, pyruvate in different compartments and so forth. And so it's a pretty complex story that's being boiled down to this one constant. And I think there might be things to be learned from trying to extract a little bit more information about what's going on. But in any case, different measures you can use, and they are arguably relatively crude. And we've been thinking about perfusion to see how I might be able to advance that. So again, I stress there's sort of lots of things that go into this curve, including perfusion, activity of the transporters, resting metabolite concentrations, et cetera. And so, if we back away from this for a second and think about, what is the simplest possible case you could look at, it's probably perfusion. And indeed, a lot of the early work in C13 focused on perfusion. And here, all these years later, we're still kind of at it. So perfusion. So this is a simplest case, and we want to see how far we can push the quantitation with this in the context of hyperpolarized C13. So this is an old subject in hyperpolarized carbon-13. This is a natural application for hyperpolarization because you don't have any endogenous background signal. You don't see anything until you inject the tracer. That's both good and bad. It means it makes your pre-scan pretty hard because you don't have any C13 until it's there. Yifan laughs because she knows how this can give you PTSD over time. The signal strength also is linear in the tracer concentration, unlike gadolinium and so forth. That's also a nice thing. And lots of tracers have been proposed. Urea, HP001, pyruvate itself can be used in a certain kind of DSC-like way to do perfusion imaging, and tert-butanol, which I'm going to be talking about today, which is an alcohol. And the reason we chose this alcohol is because it has a large distribution volume in tissue. So it basically can diffuse everywhere, whereas these other tracers over here are more or less constrained to the vascular space and a little bit the extravascular, extracellular space. So here's just to set up the contrast, or the difference, I should say, between something like T-butanol, which is diffusible, as opposed to these other traces, which are non-diffusible. Here's T-butanol in the rat brain. Here's the proton image, the carbon-13 image. You see the parenchyma enhancing here as that T-butanol gets into the tissue, versus choline, non-diffusible, where you see mainly the vasculature. So you see the bright signal around the periphery of the brain and not that much signal in the brain itself. Here's another example from the literature showing the same thing, comparison of urea with T-butanol with a nice enhancement of the T-butanol in tissue, and not so much with the urea, which cannot penetrate the blood-brain barrier. So here's another look at the T-butanol, the time course, and the two-dimensional slice, just to highlight a couple other features of this tracer. Here we've got a slice through, well, the lower part of the brain. You can see some of the arteries, the veins, and tissue here. And if you look at those three time courses, you see the arterial signal coming up quite high and coming down fairly quickly. There is some residual signal in the artery. And then you see the tissue and venous signals tracking each other very closely. And that's a sign that you really do, on a first pass of the contrast agent, you have equilibration between the blood and the tissue, and a relatively a common behavior between the blood and the tissue. And that's what makes the perfusion quantitation here a little bit easier to do. All right, so here are some images, no quantitation, but here are some images we've taken over the years in the rat heart, retrospectively ECG gated. Here's in a stroke model. You can see the perfusion defect here in this middle cerebral artery occlusion model. Here's in the rat kidney at about 1 millimeter isotropic resolution. And here are some images of the brain. We've been able to push the rat brain imaging a little bit further. And all of this was done with balanced SSFP. And I'll talk a little bit more about why we've made that choice as we go along. So the first thing we're going to do is you've got to hyperpolarize this stuff. We're going to do it by dynamic nuclear polarization on a hypersense polarizer. And basically you need to glass it and you need to freeze it with a radical. So to get it to glass, you need to make it somehow kind of a viscous solution. People sometimes use glycerol. In our case, we found that actually sucrose water combination was superior. The radicals go by these crazy trade names, OX63 and AH11501. Doesn't matter which one we use as far as we can tell. Either one of these is fine. Oftentimes you'll see people dope their samples with a little bit of gadolinium. Gadolinium has the effect of shortening the electron T1 in the DNP process and bumping up your polarization. In our case, we don't want to add the gadolinium because they're polarizing large sample volumes, and that gadolinium in our final solution is going to shorten our T1. We don't want that. It turns out that holmium DOTA does a lot of what gadolinium does. So some people help us cook some of this up. And it gives similar improvements in polarization, but it does not shorten the T1. So we use holmium DOTA rather than gadolinium. And we basically measured, polarization as a function of sucrose water, radical concentration, everything, and eventually arrived at an optimal concentration. But there's a fair amount of work that goes into this, and to really bump up your signal as high as you can get it, it does take some thought, as Ethan well knows. So the final thing was 15 millimeter OX63 or AH11501. That's the nominal concentration. People always use that. Over here, I've shown a plot of how the T1 depends on the concentration of the gadolinium or holmium DOTA, and you can see the holmium DOTA, this upper curve here. You don't see so much the suppression of the T1, so that holmium DOTA is behaving as it should. And one of the problems we always had with this sample is that when we polarize this stuff, as we increase our sample volume to the amount that we load into the polarizer, our liquid state polarization goes down and down and down. And we never could understand that. And so I went back and looked at this again, and you can see that in terms of the solid state polarization buildup, it really doesn't matter what your sample mass is, but your liquid state polarization just goes down and down and down as you increase that sample mass. And so I thought, well, if big samples are bad, why don't I try polarizing a smaller sample and dissolving it in less stuff? I actually saw the polarization go down. I thought, well, maybe if I go the other way, it'll go up. And we're a little bit surprised by this result to see the polarization go up so much as you are increasing, you're holding the concentration in the final solution constant. You're increasing your dissolution volume and increasing your sample volume that you're polarizing simultaneously. And you can get the polarization up even with these large samples and get the concentration you need in the final solution along with a higher polarization. And then the last couple of tricks were to add a magnet around the dissolution capture syringe where you're capturing the syringe as it comes out. And we can get about 34% polarization at the instant of dissolution. It's more like 18% by the time you get to injecting it. So comparable for our hardware to what you get with pyruvate. Of course, EFN can do twice that well with her polarizers with pyruvate. So that's sort of antiquated stuff. Just a quick word about The imaging methods, so I talked about the dosing. In these rats, we give about 1 ml per 100 gram of body weight. It's about 160 millimolar tert-butanol concentration. It's well tolerated. It clears it fairly rapidly. We haven't had any adverse events with this tracer. And in terms of the coils, and this is important, we've been working with a volume transmit surface receive setup. We can situate the rats with their brain centered right on this coil. They lie on their backs, and that gives us a nice positioning relative to the coil. Now, there is a coil sensitivity, and we measured that carbon-13 coil sensitivity. It took quite a while to do that in the thermal phantom. So we measured that and denoised it and so forth, and everything I'm going to show you today is actually corrected for coil sensitivity, including arterial inputs and all. But you need to be able to co-register that across studies. And so we made a 3D printed bed, and we put a little fiducial marker there, a tube with a little bit of gadolinium doped water in there. And so we have this nice ring, and when the bed and the coil move around, we can co-register our coil sensitivity on a per study basis. So that helps us with the quantitation. So that's our imaging setup. All right, so now we come to sort of the long slog of talking about how we actually do quantitation. And so just let me briefly just back up for a second and think about the class of all the agents that you could use here. So with intravascular agents, something like gadolinium or something like that is mainly intravascular in the brain, the mean transit time is going to be short. And so whatever you do, you'd better do it fast. And frequently, the washout of the bolus is overlapping with the end of the washout from tissue of the bolus is overlapping with your arterial input. And so you need to be sort of looking at both simultaneously, and you're going to have to do some kind of bolus deconvolution. There's been a huge body of work done on this, great stuff done here, but you do have to be fast. Now here, what I've plotted is the normalized signal from T-butanol in the brain as a function of time, and showing you that decay there is relatively slow. So the T1 plus flow and so forth is giving you a decay time of about 31 seconds. And so, in comparison to the intravascular agents, you've got a fairly leisurely amount of time here to do your imaging. And all the way out here to, almost 2 minutes, you're still seeing signal from this tracer. So we have a longer mean transit time, maybe tens of seconds in the normal brain when you fold in the T1 and so forth. And the kinetics in tissue are pretty simple. And so all you got to think about here is the T1, the T2, the tracer washout. And I'll just say, originally sort of the dream here was that, T-butanol shares the properties of sort of gold standard PET tracers in terms of its diffusibility and so forth. It's like 11 T-butanol or 15O water. But the problem now, of course, is we don't know a half-life, right? We don't have a fixed half-life. We have T1 and T2, and those are going to depend on what tissue you're in, what kind of imaging sequence you're using, and so on and so forth. And so I'll just wrap up this slide by saying that these long time scales are going to allow us to do relatively robust 3D imaging of the signal in tissue, and that gives us the bump in sensitivity and the ability to have good coverage and so forth. So there it is. All right, so now in terms of kinetics, I just want to stress, it's probably obvious, but the T1 is related to the flow. So normally when you measure T1 in a hyperpolarized vial or whatever, you pulse away at it, look at the decay, and that's your T1. But here you've got tracer washout as well. So you've got the arterial input, which ends at some point. We actually apply some saturation bands to shut off the arterial input. That's a nice thing about C13. You can actually kill it when you need to. And then all you've got is signal in tissue and the signal in the veins. And the thing about the diffusible tracers is that, as I mentioned before, the tissue and the veins are basically in equilibrium. I guess I should have used the same shade of blue here to stress that. They're in equilibrium up to an overall constant, which is this partition coefficient. I called it lambda here. I've defined it as the ratio in equilibrium of the tracer's concentration in blood, in brain relative to blood. And it comes out about 1.16, albeit with a large error bar from some direct measurements. And these are the folks who measured that. Now, if the T1 were infinite, then the measurement of the decay would just be outflow and you'd be done. Okay, so T1 is flow. But when you include the T1, of course, you have an effective T1, which is both the ordinary R1 from just the ordinary NMR relaxation, plus the flow divided by this partition coefficient. And so you can relate in various ways the T1 and the flow. So imaging. Whoops, did I get, okay. Where am I? Okay. So we need a rapid imaging sequence. It doesn't have to necessarily be a fast sequence, but rapid. So we use balanced SSFP. It's a natural choice. It has high sensitivity. The magnetization in the BSSFP steady state tends to decay more slowly. It's not a T2 star decay. It's not a T2 star decay, but rather a T2 decay, which extends the lifetime of the magnetization. Although it's like a gradient echo sequence, as we know from the work of Scheffler and others, it has spin echo-like sequences. The magnetization gets refocused at every TE. And so you have this nice slow decay. The drawback of balanced SSFP is the off-resonance sensitivity. But we're working with the nucleus carbon-13 where the gyromagnetic ratio is down by a factor of 4. So even at 9.4 Tesla, we can live with that off-resonance sensitivity. And if you include the RF pulses and the flow and everything, you end up with an expression like this for the effective rate of decay. Of course, it now depends on the tip angle. As you go up on the tip angle, you see more T2 decay, and that's a fairly rapid decay. As you go down on tip angle, you get into a more T1 dominated phase where you're seeing just a T1 plus flow. And so obviously by varying this tip angle, we can get at the relative contributions of R1 and R2. All right, now the other thing that we're going to need is we're going to need an arterial input, and there the challenges are a little bit different. The arterial input is probably better to do 2D, and so we're going to try a hybrid 2D, 3D strategy to get the arterial input followed by the tissue signal. And the reason we're going to try the arterial input 2D is because you just get the spatial and temporal resolution that you need. If we were to look at the arterial input with the 3D scan, it's hard even to pick up, and I'll show you that we do pretty well with this 2D sequence. Now, there are some caveats to the arterial input, which of course is partial volume, the rapid flow can distort the signal and so forth. And so we're not going to rely on the arterial input function to measure flow. There's a couple of issues there, these that I've highlighted here already, but there's also, of course, the overall normalization of the 2D relative to the 3D. You're acquiring them with different sequences. And so you need to think about that as well. And so we're going to use T1 and various trends with a flow with T1 with flow to try to pin down the signal. The flow, I should say. So here's what this 2D, 3D imaging looks like. For about 17 seconds, as we're injecting the bolus, we monitor the arterial input, which looks something like this. So you see the arteries here lighting up and then a little bit the veins. We apply sat bands below the neck to shut off the magnetization that might arrive late. So we believe that we're getting the whole arterial input, all of it, start to finish. We have delay of about 9 seconds, which is basically a hardware delay on the magnet. And then we do 3D mapping of T1 and T2 for about 30 seconds. We let it run for 60 seconds in case there's any signal out there, and I'll show that there is a little bit. And so basically it's arterial input, delay, map of the signal amplitude, T1 and T2. All right, so basically you got this R1 effective and R2 effective. R1 effective is the tip angle independent part, and you've got a tip angle dependent part. And so we're going to map this just by varying our tip angle. Now the residue function, that is to say the decay of the signal in tissue should be well approximated by an exponential, and it's pretty easy to work out that in terms of your arterial magnetization and your tissue magnetization, you have some convolution with an exponential decay. And so you get to an expression for flow, which is basically the signal amplitude in the signal. The brighter the pixel is, the more flow you've got, normalized by the arterial input, suitably weighted by sort of the amount of decay that has happened between when the magnetization arrives in tissue and when you image it. And so that's why we have this convolution with the exponential down here. Now I put a factor kappa in here because the denominator here is 2D, the numerator is 3D, and we've got all those normalization issues and so forth to think about. And so we're going to normalize using an alternative strategy rather than trying to just get the AIF. All right, or excuse me, get the absolute normalization of the AIF. So T1 and T2 mapping. So we basically, as I said, we vary the tip angle, and the basic idea here is we walk it into a steady state, and for about 20 seconds, we image at a 4-degree tip angle where the magnetization decays at a rate dominated by T1. There's about a 10% T2 contribution in gray matter. Then we transition with 32 pulses up to a 24-degree tip angle steady state, and we image for another 34 seconds. And now the decay is much more rapid, and we don't get a lot of signal, that far out, but we look at it all anyway. Yeah. All right, so I'm going to talk about a little validation study that we did here. This is 8 Fisher rats imaged both at baseline and a second time with microsphere perfusion quantitation. I'm not going to talk about microspheres today in the interest of time, but it does bear on some of the data we've got. This is all done on a Brooker Biospec 9420. It would be wonderful to have a 4.7. We don't have a 4.7 anymore. The T1s and T2s are a lot more favorable there. And this old-fashioned Oxford hypersense polarizer. Imaging protocol, proton anatomic imaging, dual gradient B0 field map plus a map shim to shim from the carotid arteries where we're getting the AIF all the way up through the brain. This also gives us our co-registration for the coil sensitivity. And then we do our carbon-13 imaging with the 2D, 3D balances SFP. And I'll just mention briefly that for the baseline scans, before we do the microspheres and everything, we run at 1.2 millimeter voxels on all 3 sides. 1.2 by 1.2 by 1.2. For the sphere studies where we have a little bit of reduction in flow, we were a little worried about SNR, and so we went up to 1.5 millimeters cubed for that. And the sphere, the animals are sort of prepped for the sphere infusion before they go into the magnet. As soon as we're done with the scan, we bring them out, we infuse the spheres, and then we can, of course, euthanize and harvest tissue to send these spheres back. For any of you who are working on this, there's a non-radioactive version of microspheres we've been using. There's a place out in Worcester that can actually make the spheres radioactive after you harvest the tissue and count them that way. So you don't have to deal with radiation in the lab. And I'll just say briefly that to infuse these spheres, you have to get the spheres somehow into the left ventricle. And in order to do that, our very talented surgeon, Mauricio Contreras, worked out a way of threading a catheter down through the right carotid artery, into the heart, into the left ventricle. And the only reason that's relevant today is because in so doing, he ligates the right common carotid artery. And so what this gives us is it gives us a perfusion knockdown in one hemisphere of the brain. And so we can actually look at normal and ischemic brains in the context of this study. And I'll show you how the ischemia impacts the imaging. All right, so here's what the 4-degree tip angle images look like going down the stack, sort of every other frame. Now you see when you go up to 24 degrees, you have pretty rapid signal dropout, particularly in regions of white matter, but able to see some signal out for several frames yet. And so this sort of rapid signal decay that we're seeing in some of these white matter regions is a challenge, and I'll talk a little about that as we go on. All right, so here is a baseline study. So here's the proton image in the rat brain. Here is the normalized signal amplitude. So we fit to the initial signal amplitude, which is that amplitude that appears in our expression for flow. And then we look at how the signal decays during the low tip angle phase. We ramp the tip angle up to 24 degrees, and then you keep on, you track the decay during that high tip angle phase. So here, there's some cortical gray matter shown here, and here's what the fit looks like. We've got caudate putamen here. That's what it looks like there. Pixel in the thalamus looks something like that. And lastly, I put a pixel in the brainstem, which I can't, here it is here. And you see the very rapid decay that you have in the white matter of the brainstem. So that's sort of a challenge that we have. And I can say more later about why we think that might be happening. But here's the amplitude, R1 effective, R2 effective. You see these spikes probably related to the white matter. You see the R squared of the fit. It's pretty good, maybe a little too good. I'm going to have another look at that and make sure that that's right. But the R squared is pretty good. And you see the points in the curve seem to pretty much align. I'll also note that, of course, you have the lower decay rate, 0.055 per second in the low tip angle phase, and then the tip angle, and then the decay rate ramps up as you ramp up the tip angle. So here's an example now in the ischemic state, and you see this knockdown in the amplitude here related to this perfusion defect from ligating one of the carotid arteries. And we would expect to see that also in the R1 effective map because there's going to be reduced washout. And so we see that again here. R2 is largely unchanged. I'll show you that quantitatively in a moment. Here's the R squared. And I'll just note here, we'll compare the pixel A and B in the low flow or high flow hemispheres, and you see the very, very slow decay that we're observing over here on the low flow side. It's taking about 50 seconds as the time constant of the washout. And so quite slow washout in this low flow state relative to what we were seeing before, which is sort of 0.05, 0.06, something like that. And then down here again, you see in this sagittal view, this spike in the T2 related to those white matter tracks. All right. So what's going on in the white matter? We just wanted to go back and validate this with some 2D imaging to make sure that the T2 really is that short and get a handle on what's going on there. Here is a sagittal slice. We imaged this 2D. You see the very rapid decay of the signal in the white matter here in the brainstem. Put ROIs on it, and you see here this shortest decay curve with a time constant of about 0.6 seconds is the white matter. This was done again with balance SSFP, factoring in the tip angle and so forth. You end up with sort of a 60 or 70 millisecond T2 in the white matter for this tracer. So that's one sort of challenging thing that we're dealing with. In any case, we use this to constrain our fits in the 3D data because we really don't have enough temporal resolution to probe that short of relaxation time very accurately. So here, just to show you, that we actually can measure things now. So here are the R1 and R2 values measured in different ROIs around the brain cortex, putamen, thalamus, hippocampus, brainstem, and so forth. And here they are as box and whisker plots. These three groups of ROIs here, all n equals 8, mean plus minus standard deviation, and so forth. These are not statistically significant from one another. but these are statistically significantly different from the lower values. In terms of the R2, you see a much wider distribution, and you see the brainstem white matter standing out way above the others with this sort of 60 millisecond T2. And all of these are statistically significant, all these differences here. So that's what it looks like in the normal brain. Now, stepping away from the whole brain and just looking at some cortical gray matter here, we can look at what's going on with the R1 effective in the normal and the ischemic brains. So here's what the R1 effective is in cortical gray matter for a baseline study. Then you look at both the high flow hemisphere and the low flow hemisphere in the cases where we've done this unilateral carotid ligation. And you see the R1 effective stepping down as you would expect, and that's flow. Now, in proton imaging, you're not going to see, much change in the proton T1 and T2 in the, acute phase of ischemia in the brain, right? That's why we do diffusion and things like that. And so the question is, if you look now at R2 effective, which should be independent of flow, here you just don't see any change at all. There's no significant change in the R2. Of course, the R2 is quite fast. So in any case, the overall pattern that you expect is holding up. So now we're going to actually try to extract the flow. So now I've got this signal amplitude, which is here. And we've got, basically a T1 corrected AIF. We've got an overall unknown constant here, which we're going to treat as just a normalization. Here is a slice where you measure the arterial input. You see the two vessels here on proton and again on C13. Here's the arterial input. At the end of this sequence of scans, we apply SAT bands and shut off that inflow. And here's the integral and so forth. And you put all this together and you get a map of relative flow. Okay, so some hot spots back here. I believe this might be the superior colliculus. We were a little surprised to see such high flow in the brainstem as well, and they're following up on the literature to see if that's real or if that's somehow related to an artifact of our imaging. But in any case, that's what your relative flow looks like. Here it is again in the ischemic case, and you see this really dramatic knockdown in the relative flow. In this case, this is the case that showed the most dramatic knockdown. Here down in the sagittal slices, we've shown a slice which is offset a little bit toward the ischemic side of the brain, and you see the perfusion defect here on the forebrain. So that's what it looks like in an ischemic case. So now let's put in the last ingredient here, which is the flow normalizations. So we have all these sort of relationships that we can use to try to tack down the relationship between the R1 effective and the flow. And there's a couple of different approaches that you could use. So our absolute flow, the thing we actually want to get is some constant multiplied by this relative flow that we measure from the imaging. And the things we don't know are we know the R1 effective, but we don't know sort of the underlying R1 of this stuff in tissue. We don't know the absolute flow, but we know it's proportional to the relative flow, right? So if the T1 is independent of flow, and again, in the human brain, in ischemic stroke and so forth, the T1 isn't changed much by that kind of acute ischemia. If the R1 effective is linear in flow, and the T1, the R1 effective will be linear in flow if T1 is independent of flow, and we'll talk about that in a minute. We also know the slope of this line, which is going to be the inverse partition coefficient, and we also know the intercept, and we can extract that from fitting using just the relative flow. So if the R1 is known anywhere, if the T1 is known in any kind of normal tissue, then in that tissue you can put an ROI there, measure the R1 effective, you know the R1, and that tells you the absolute flow. Now that you know the absolute flow in some little ROI where the T1 is known, you can turn that around, get your kappa, and you know the flow everywhere in the image, right? So from one pixel, one ROI, you can normalize the flow. I'm going to argue maybe that's not the best way to do it, but it was tempting. Alternatively, we can use the slope of R1 effective as a function of relative flow to determine what kappa is. So we know what the slope should be, and so we can use that to fix this normalization constant. So here, I drew a lot of ROIs. Here is a plot showing all of the normal brains and all of the ischemic brains versus relative flow. Most of the normal animals are sort of what you see here is we're expecting a linear trend. And we see it up here reasonably well, but then when you get to the most ischemic cases, there appears to be some roll-off. And so this assumption that we've made, or that we would like to make, that the T1 is independent of flow probably doesn't hold up. So if you throw away, just sort of arbitrarily as a judgment call, if you throw away everything below this line, which appears to be sort of the knee of that plot, you can get a couple of different plots here. So here is in blue, That is the case, including the ischemic cases. You get a little bit steeper slope, and you get a little bit longer T1. So this 0.022 here is the inverse T1. And, I looked at this a lot of different ways, and you do see that intercept, which is over here, that's telling you the T1, appears to depend on the degree of ischemia. So I trust this red line here a little bit more, which has a little bit less slope, has a little bit shorter T1. So now you're at about a 35 second T1, something like that. And you have some constant that is telling you, that gives you the slope in terms of relative flow. And we know that the relative flow, when we replace that with absolute flow, that this line should have a slope of 1 over lambda. Okay, 1 over the partition coefficient, basically 1. And so doing that, we figure out what that normalization constant is. A normalization constant says the 3D amplitude is a lot bigger than the 2D single amplitude. And that's what you expect. 3D should be bigger than 2D. And here it is in terms of flow, in terms of milliliters per milliliters per second. So it's a pure rate. Here it is again in terms of milliliters per 100 grams per minute. And that is your T1, 30, 35 seconds, something like that. And this is your slope. And so now with this normalization constant kappa, we can go through normalized flow and get flow in milliliters per 100 grams per minute. And so it looks something like this. Here's that study I showed you before, arterial input and so forth. And here's the normalized flow in milliliters per 100 grams per minute. And I don't have all the ROI analysis and everything, but wanted to show you that it's here. Again, we see this hot spot, maybe in the cerebellum, maybe superior colliculus. We spent a lot of time looking at that. It appears to be what this model predicts. So we'll need to continue to think about that. But over here, normalized with the, all the information, that is the normalized flow here in this normal brain. And for the ROI in the top right, your flow is about 160 mils per 100 grams per minute. That's a little bit on the high end of the ASL literature, but it's at least in the ballpark. Here it is in this ischemic brain. And you see that we're able to pick up this signal. Comparing these two ROIs here on the flow map, of course, there's a knockdown in flow overall, but also a knockdown, dramatically on the left-hand side of this image. And we're picking up a flow of about 28 mils per 100 grams per minute on that side in that ischemic brain. So, sensitivity down to relatively low levels of flow. So that, kind of a long slog to get there, but I think that we can use the properties of diffusible contrast to get a fairly robust normalization of the flow. And we can, detect flow in these ischemic models and so forth. And so that's sort of where it is. All right, so I wanted to say a few words about CSF because as you get out to the later time frames, there is flux of this tracer into the CSF, and we're not entirely sure what this means. And so if people have any comments, I'd love to hear it. So here's stepping through the slices of the later frames, and you see in all these regions of CSF, you see the bright signal of the carbon-13. Here's another example showing this going out to about a minute or so. And here in this little pocket of CSF, you see the bright signal here in the CSF. Here's what the time courses look like, not normalized in any particular way, but you see this bright signal in CSF out at these later frames. So it's there again and again and again. What are the time frames associated with this? So we went into the T2 weighted images. We tried to get some CSF enriched pixels by just picking out the long T2 pixels here highlighted in red. Put that mask onto the C13 images, and you get these points. Okay, so the points are what we actually measure. If you try to fit that to a single exponential, you get a decay time constant of 3.3 seconds. I should say these are the late frames, the high tip angle, 24-degree tip angle frames. You've got the time in seconds down here, and you don't get a great fit with a single exponential decay. If you add a second exponential and let the parameters drift around, you find a short time decay constant about 2.7 seconds that dominates earlier here. And now at the later time frames, you get sort of a 17-second, quite a long time constant for the decay in this sort of CSF-dominated regime. If you compare that to if you try to translate that into a T2, which you can't do entirely, but you correct for tip angle and so forth, corresponds to, I believe, a T2 in 1 or 2 seconds, something like that. So quite a bit longer than the T2s that we've been observing. So, generally in the gray matter and so forth, it's sort of 300 to 500 millisecond T2s, and we're seeing this much longer T2 in the CSF. So any comments would be welcome. All right, so now if we forget about the quantitation and just say how far can we push this in terms of image resolution, here's some images that we got at just under about a 0.5 millimeter isotropic resolution using these 3D methods. Scan gets a little bit long. It's about 13 seconds per frame. So basically you burned up all your magnetization at the end of the first shot. But here you see the kind of images that you can get. These are the carbon-13 images, not the proton images. And you see that you can now finally sort of begin to see the CSF even very early on in the lateral ventricles and lots of other detail. And I should get a neurologist up here to talk about all the things you're seeing. And here's what it looks like in terms of a movie showing all the slices. And one of the challenges you run into here is sort of the T2 blur. That is to say, during this 13-second acquisition, you have to acquire these centric because there's quite a bit of decay as you get out to the edges of K-space. What else was I going to say about this? And if you look at the other planes, these were acquired coronal. If you look at the axial and sagittal planes, they aren't quite as clean because of that T2 blur, but these are the sort of the most visually striking ones. And now, in the last few minutes, got about 5 minutes left, I want to talk about how we've been trying to push these balanced SSFP methods into carbon-13. And this stuff goes back a long way. I was looking at ways to do this with pyruvate years and years ago, and sort of got distracted by other things, but I just wanted to put a few pulse sequences out there. So we'd like to do balanced SSFP, but we'd like to do it spectroscopically selective. And so there is this workhorse sequence, wonderful work done at UCSF, using basically a spectrally selective excitation followed by a spiral readout. And so they basically excite a single metabolite at a time and read it out with this spiral, and they can get these nice images. So they've got these images here in a patient with a renal cell carcinoma. You can see the high lactate detected with this method. And so really a nice sequence. But there's a couple of things about this that I would sort of like to get around. The first is the RF is pretty long. It's about 9 milliseconds, and your TR is even longer. It's about 15.3 milliseconds. And those of you who worked with balanced SSFP know that even on carbon-13, that long of a TR, you're gonna have to shim pretty carefully. So what else can we do? So here's some work I did with Gopal Varma quite a while ago, where we try exciting all the metabolites at once and then use this sort of balanced SSFP profile. So as a function of off resonance or as a function of your phase advance, you have different amounts of signal coming in balanced SSFP. So usually you image at a phase advance of let's call it 0, and you're sort of on the sweet spot of the balanced SSFP, and you don't have these bright bands. So these bright bands represent the the sort of blow up in the signal as you have more off resonance. And you can place your metabolites across at different points on this curve. And by acquiring at a couple of different phase advances, you can get different linear combinations of those metabolites and pull out pyruvate, lactate, alanine, and so forth. And so here's an example we did in a renal cell carcinomas, A498 cell line, as I recall, a little bit of off resonance there. And we compared the EPSI at 2.5 millimeter in plane resolution with the balanced SSFP at 1.25 millimeters in plane resolution. And you see you pick up the pyruvate and the shape of the tumor, maybe a little bit better with the balanced SSFP. You see the lactate signal. Here's the pyruvate hydrate sort of blown up by a factor of 10. And it was sort of a signature of sort of accurate selectivity that pyruvate and pyruvate hydrate had a similar distribution. One challenge you ran into here, of course, is the off-resonance sensitivity. So you're bringing more and more bright bands into your acquisition. And so this off-resonance over here shown in this field map probably gave us a little bit of this banding artifact out here at the edges. Here's another sequence that we tried where we tried doing fewer phase advances. So not sampling along that balanced SSFP signal response curve so much, but doing 2 phase advances with 3 echoes per TR. So of course, acquiring several echoes will give you different phases for the different metabolites and use that to extrapolate, to extract the different metabolite signals. And so here's some stuff we showed at ISMRM, quite a while ago. Here is an early image acquired in a tumor, a mouse tumor, about 14 seconds, see the pyruvate lighting up and quite a bit less signal in the alanine, pyruvate, and lactate channels. You wait out to about a minute, you pick up the lactate signal as you expect. This is at about a 1 millimeter squared in plane and 4 millimeter slice acquired 2D. One of the things you run into here actually in the balanced SSFP is that your slice select, you tend to run out of gradient strength just selecting your slices on carbon-13. And so that's why the slice is so thick. So we'd like to advance this into a 3D sequence, probably a variant of this. There is one last thing that I'll show you, something I've been working on lately, quite in preliminary stages, but with just 3 echoes, if you choose your TR correctly, in these tumors, you generally have 4 metabolites you're interested in, which is pyruvate, pyruvate hydrate, alanine, and lactate. But the pyruvate and the pyruvate hydrate basically track each other. And if you make the appropriate choice of TR, you can get that balanced SSFP profile set up in such a way that pyruvate and pyruvate hydrate basically collapse into the same channel. And so now all I have to worry about is the alanine and the lactate. And so with this choice of TR and some other things worked up in sequence, here are some images acquired in a rat early on just to check the spectroscopic selectivity of it. Here's the rat kidneys. We inject pyruvate. And here's what we get for a pyruvate plus pyruvate hydrate image with some coil sensitivity in there. And we managed to reject the signal in the lactate and alanine channels. And so there's some selectivity. This was done with a 2 millimeter in plane. And again, this is something I'd like to generalize to 3 dimensions. So, this is something that the oncologists are pretty interested in. They're interested in, response and resistance to targeted therapy in cancer. And so something that Leo Tsai and Gerhard Wolf over at Beth Israel Deaconess have been bugging me about for years is to be able to look at, within the tumor, to be able to look at the heterogeneity. And I think, the hope is that some of these high resolution imaging techniques, together with some of the quantitative imaging techniques, might enable us to begin to do that. And the reason they want to look at these things in high resolution is that as a tumor acquires resistance to a targeted therapy, it's probably not the whole tumor that goes all at once. It probably have islands where the genetic mutation, where the tumor begins to develop resistance. And then over time, if you can follow that, that would give you, a sign that, the resistance is developing and you need to start looking for another therapy. And so clinically, Gerhard Wolf is pretty keen on being able to do that. So let me just wrap up. Okay, so I've shown, a pretty long slog to get there, but with the T1 and T2 mapping and measuring the arterial input, we do reasonably well in this quantitative 3D perfusion imaging, at least in the forebrain. Some details in the hindbrain we need to think about. You got sensitivity in these rats down to about 20 mils per 100 grams per minute with pretty good SNR. I think we could go lower. Sort of the raw C13 SNR before you do any processing is about 20 or 30 to 1. And so you can trade that for resolution or sensitivity to low flow. As I said, the quantitation is straightforward but arduous, and so we need Fenn and Matt Rosen and others to cook us up a tracer with a longer T1, right? If you had a T1 of a couple of minutes, you wouldn't be worried about any of that. You just measure at a low tip angle and you would be done, right? And T1 would give you flow. We'd like to explore this, co-polarized with pyruvate so we can get perfusion and metabolism at the same time. We'd like to push these BSFP techniques into pyruvate metabolism. And, just as one closing thought for something for, Yifen and Matt Rosen and so forth to think about, might be interesting to co-polarize all three, polarize pyruvate, urea, T-butanol at once to sort of span the sort of range from a more intravascular, partially permeable agent all the way out to a freely diffusible agent, pyruvate somewhere in between, as a way to be able to look at things like, active transport and efflux of the lactate in and out of the tissue. So I'll close now. Very special thanks to Gopal Varma, the world's most patient collaborator. Cody Callahan was in my lab for many years, helped out with a lot of this. Mauricio Contreras is the surgeon without whom those ischemia studies have been totally impossible. Lots of people have contributed over the years. These are some of the folks in cancer biology have worked with us over the years, and I'd like to thank funding as well. So with that, I'll close, have a sip of water and take some questions. Thank you.